Okay, welcome back to the second day of the workshop. So, yesterday we saw a nice introduction to the idea of non-genetic heterogeneity. So, remember that for a very long time, people thought that resistance in cancer is driven purely by mutations in the genome, right? And this idea slowly has evolved, and right now what we think is that often, at least in the initial phases, you might be overcoming the effects of drug simply because of non-genetic fluctuations, right? So you can have fluctuations in gene expression, you can have differences in the epigenome. These lead to cells that are sort of primed to avoid the effects of the drug. Okay, so this is, you saw these things very beautifully introduced yesterday in both the set of lectures, both by Yogesh and by Nandini, right? And the other important thing that Yogesh introduced yesterday is the idea of, the cell state switching and these, what he calls these twin experiments, right? So if you remember what he was talking about yesterday is that if you have barcodes in these single cells, then what you can do is you can track clones of cells and look at these twin experiments and try to figure out when this non-genetic heterogeneity actually arises. And he showed that in his data and data from other labs, for example, Arjun Rajas, and in fact, there are other people who've done this even before. There's a very nice paper from Nature Medicine from back in 2013, I think, Bhang et al., where they also do these kinds of twin studies with barcodes. And you can see that, if you remember this very nice slide yesterday where you saw that on the diagonal, most of the points lay on the diagonal, right? So, which shows that resistance that comes up, the barcodes that come up in twin 1 are also the same barcodes that come up in twin 2. Okay, so you see dot points that lie along the diagonal. And this suggests that these non-genetic states are in fact mostly occurring before you even add the drug. Okay, so this is a very interesting conceptual advance that has happened in the last 5-10 years. The idea that a lot of this non-genetic heterogeneity actually exists well before you even add the drug, okay? And what, if you remember Yogesh's talk, what he ended with is this idea that you want to now quantify the dynamics of these non-genetic states, how they switch, on what timescales they switch. And he introduced this term called memory genes, where he says that, this idea comes really from Sidney Schaffer's paper, where where you argue that if this time scale of switching these states is slower than that of a cell cycle, then these are called memory genes, right? So you inherit this state across a few cell cycles, okay? So quantifying the dynamics of these states is now a very interesting open problem. And there are a few ways in which people have already attempted it. And so today what I'm going to show you is one way which utilizes recent developments in imaging techniques. So these are single cell imaging studies where you can actually track single cells over time. So that's what this cartoon is meant to represent. You have single cells, and each snapshot is a moment in time. Okay, so you can use microscopy to track these single cells, and you can infer, or you can actually measure the relationship between each individual cell. Okay, so notice that this is at a much higher resolution than what Yogesh was talking about yesterday. So these barcoding studies allow you to look at clones where you have, let's say you have 10,000 cells, all of which have the same barcode, okay? But you don't know the relationship between each individual cell. You know that all these 10,000 cells come from some common ancestor, but the intricate relationship between these cells, like these two cells came from the same grandparent, for example, in this case, these are things that you don't know in barcoding studies. Right? So these studies give you an advantage of very high resolution at the single cell level, okay? So these things can be used, and I'll show you how in estimating the dynamics. So before we start there, I want to show you a very simple movie. This is a movie I generated while I was at the end of my postdoc, and you'll see something really interesting even in this short sort of 30-second movie, okay? So these are two cells whose nuclei are stained with a particular reporter. This is an H2B reporter. So you see only the nuclei of these two cells. Now let me play this movie, and I want you to see something really interesting that goes on. So you'll see that this single cell divides, and now you have two daughter cells, okay? So these are my two daughters. Now I want you to watch what happens to these two daughters in just a moment, okay? You'll see something really interesting that happens. So you see now that these two daughters are sort of separating in space. They are moving apart, and there, right? What you may have noticed is that the two daughters of this cell divided in the same time frame, okay? So that's quite remarkable, because the daughters of these two cells were separating in space, yet somehow they had information about the cell cycle times of each other and divided in exactly the same frame, okay? So each frame is half an hour, so there's of course an error of about half an hour here, but within that error, they divided in the same frame, suggesting that when you have lineage relationships, remember those two cells were daughter cells arising from the same mother. So when you have lineage relationships, usually you tend to see correlations, okay? So those two cell cycle times were strongly correlated. It turns out that this is an extremely ubiquitous phenomenon, and in many different contexts, you start seeing lineage correlations show up. So these are two different examples. This is from a work that we did a few years back, and you can see here that these are individual traces of single cells. So the way to look at this is that you have some parent cell here, and these are the lineages of these singles of this parent cell, and each track here tracks the temporal evolution of a fluorescent reporter that we had in this system. In this case, it was P53, okay? So P53 protein was being tracked along this line, and that's this row over here, okay? This row here is the sister cell that is over here. And what you can immediately see without doing any quantification, just visually, is that you can see clustering, right? You can see that when you are close by in lineage space, you tend to see the same kind of dynamics. So red here indicates cells that die, blue indicates cells that survive in response to a drug that was added at this time point. And you can clearly see that the behavior is strongly correlated, yeah? So if you are close by, these are all red, this chunk is all blue, this chunk is red, okay? But if you watch closely, what you'll see is there are interesting places where you see that sisters may not have the same fate, okay? So for example, I think there was one case here, for example, this one, you have two sisters where one sister is red, one sister is blue, okay? This suggests that even if you have strong correlations, which indicates some kind of memory and inheritance in your system, you also have cell state switching, okay? In these two sisters, you see two different outcomes. Similarly, if you're not looking at dynamics, but just mRNA expression, and this is in fact experiments very recently done by Vishnupriya, who's sitting at the back, she's in my lab. This is just gene expression, and you're looking at gene expression of two sisters after doing time-lapse microscopy of a particular gene, you once again see strong correlations, okay? So almost any quantity that you measure in lineage-related cells tends to have strong correlations, okay? And so then the question, and this is of course not a new phenomenon, people knew this from a while back, but what is becoming very interesting is the question of how do you use these lineage correlations to infer dynamics of these cell states, right? Because you can imagine intuitively that there is information of the dynamics of cell state switching in these kinds of correlations. Why is that? If the switching was very fast, just imagine that these cell states are switching very rapidly between each other, then what will happen is, and this switching is faster than the time scales of a cell division, then what you'll have is completely uncorrelated lineages, right? Because you are changing between states extremely rapidly. But if the switching is slower, and this relates back to what Yogesh was talking about yesterday, memory genes, if you have such memory genes in your system, then what you expect are correlations of this nature, okay? Or correlations of this nature. And then the question is, in the lab, you can measure such correlations. It's hard to measure dynamics like this all the time. So from these kinds of correlations, can you then back out what the underlying dynamics must be like? Okay, and what I'm going to do is talk to you about this really nice paper that came out of Michael Elowitz and Boris Schreiman's lab some years back. It's in Cell Systems, you can take a look. I'm only going to be able to discuss one small part of it. The rest of the paper is also very interesting, but this is what we are going to try to finish with by the end of today's class, okay? This paper where I think for the first time, I mean, before this, I'm not aware of approaches that use correlations to infer cell state transitions, okay? Any questions here? So this is broadly what we are going to cover, okay? The biology. So you have these single cells, you have time-lapse microscopy data, you have measurements of cell states using some kind of gene expression measurements. In this case, this is going to be single molecule FISH, okay? So where you can measure gene expression of single cells, you'll have correlations at the end of this lineage tree. So you are able to measure the cell states only at the end, okay? Because when you do these measurements of gene expression, you have to kill the cells. So at this time point only, you are able to measure the cell states. And so from that static snapshot, you want to infer dynamics. So conceptually also, this is very interesting because you only have a static snapshot. That's the point where you fix the cells and did your single molecule FISH, right? And from that static snapshot, you want to extract dynamics. So this again is a very nice conceptual approach, and my lab also has been working a lot on these kinds of ideas, but today I'm going to cover only this. Yeah? Okay, good. So at this point, I'm going to stop the slides, and the rest of the talk is going to be on the blackboard. Okay, so we are going to see how you can even approach these kinds of problems. And the idea is what we want to do today is develop a theory, which is theory of Markov chains. You'll see how Markov chains are used to model biological systems. You'll see some very interesting properties of Markov chains. For example, these long time, what are called stationary distributions. And using these properties, you'll see how you can use data like this to actually rigorously infer dynamics of cell states. Okay? So, yeah. Sorry, say that again. Yeah. No, so forget the, even I don't know what this is. Vishnupriya knows, but that's not important for the purposes of this talk. Just each dot here represents the gene expression of one sister cell and the second sister cell. That's all that you need to know for this talk. Yeah. Any other questions? Yeah.
So there's an alternate technique, which was, I mean, I know of it, a paper from the University of Pennsylvania on cancer, where they have a what they call programmable lineage tracing system.
Right.
Maybe offline you can tell us about what you think about that.
So I'm not familiar with the one from UPenn, but when you say programmable, these are the ones that can introduce mutations over time. Right. So in fact, Sahan
originally was done in the developmental system, and there was a cancer biology.
Absolutely, right. So what Herbie is referring to are these barcoding techniques, but with the limitation with barcoding techniques is that once you introduce the barcodes, they don't themselves evolve over time, right? So once a single cell has a barcode, all its progeny will have the same barcode. But this is not coupled to the cell cycle, so you can't resolve later cells and say which one is sister, which one is cousin, and so on. But what Herbie is talking about is programmable barcodes, where over a certain number of divisions, you can introduce new mutations into those old barcodes, right? So you have some sort of a timer, and then at the end, you get much more highly resolved data. But the problem with these things still is that they are not tightly coupled to the cell cycle. So they happen stochastically every, say, 3 or 4 cell cycles. So you still don't have the kind of resolution that you would want to get sisters, cousins, first cousins, and so on. So in fact, there are a number of things. In the same group, Michael Elowitz's group with Sahand as one of the first authors, they also have a technique, it's called memoir, where they do these kinds of scratch pad assays where you introduce new mutations. Int memoir, exactly, right. But none of these techniques currently are able to strongly couple it to the cell cycle, okay? Yeah.
I wanted to ask, the idea that I got from your talk was you're converting from...
I haven't even started my talk.
Yeah. I know, but like that you're converting from static to dynamic, right?
Right.
Why can't we just like track cells, like building a tracker and then...
Okay, fair point. So the reason why in most cases you can't track cells is because it's very hard to develop live cell reporters of gene expression. Okay, so that's a very hard technique. Even if you could track perhaps 2 or 3 at max, because you're limited by the number of colors that you can image. You have filters, right? So, but here, the good thing is at this endpoint, with the current kind of technologies that we have in single molecule fish, for example, you can fish for 10,000 genes, right? So these are much more high throughput and allow you to actually infer these cell states. Okay, so okay, I should move on to the board because we have a lot to cover. So I don't know if the tech team is here. Can we move this thing up? Okay. So now we'll move on to the board, and since most of you said that you have some background, I'll just very quickly introduce you to certain important ideas which you may not have seen before, and that will be very important for the rest of the talk, okay? So first is conditional probability. So can you all see the writing at the back? Yeah, this is big enough? Okay, good. So let me just see how. So can someone tell me what this quantity is equal to in terms of the joint probabilities and so on? Okay, P joint probability of A comma B divided by P of B. Is this okay with everyone? Is there someone who hasn't seen this before? Can I assume that all of you have seen this? Okay, no one is saying anything, so I'm going to assume that you've all seen this. Okay, so this is one, and from this you have the important idea of independence. So if P A intersection B is PA, right, then you have PAB is equal to PA, PB, okay? This is what is called independence, right? So if you have 2 random variables, A and B, which satisfy this over here, where the joint, where the conditional probability is just this quantity here, then A and B are random variables that are independent, yeah? So this will also be important coming up. Okay. So that should be familiar to everyone. So the next thing that we'll need is something called the chain rule. So it's basically, I want to extend this to an arbitrary joint distribution. So we'll start with a joint distribution of 3 random variables. And what I want to do is, I want to write this in terms of conditional probabilities. And you'll see that this becomes very important in problems like this lineage inference of kinetics. Okay? So this, if I just use this quantity here, sorry, this number 1, expression number 1 here, I can write this as P A comma B, P C given A comma B. Okay, so I've just used 1 here and I've clubbed A and B together. Okay, so this should also be very simple following from this. And then I'm going to use 1 once again to break this down. So I'm going to get P of A, P of B given A, P of C given A comma B. Okay? So this is very simple, but this is incredibly important. Okay? And in fact, this kind of breaking down of a joint distribution lies at the heart of many powerful techniques such as graphs, right? Graphical modeling is completely based on representations of joint distributions in this kind of form. Okay? So this might look super simple, but it's actually very useful. So if I just generalize this kind of simple chain rule, what you'll get is generalizing. What you'll get is, now if I call my random variables with alphas, alpha 1, alpha 2, all the way up to say alpha k is P of alpha 1, okay? Then P of alpha 2 given alpha 1, P of alpha 3 given alpha 1, alpha 2, and so on. Okay, all the way to the final term, which will be P of alpha k given alpha 1, alpha 2, all the way up to alpha k minus 1. Okay, so that is the most general form of what is called the chain rule. Okay, I'll call this equation 2. Very simple, but very, very useful things coming up in everything that we are going to do. Okay, so that was the chain rule. And finally, the other thing that we'll need for an introduction to Markov chains is something called the law of total probability. So this is also very intuitive, as you'll see in just a bit. So I'm not going to derive it in that sense, but I'll write this down, what this law says, because once again, this is going to be very useful. So what it says is, let B1, B2, all the way up to Bn be a sequence of events such that the following properties are satisfied. P of bk is greater than 0 for all k, okay? And B, Bi and Bj are disjoint. Okay, so there's no overlap between the events B1, B2 or B2, B3 and so on when I not equal to j. Okay, and these two properties together is what is called a partition of S. Partition of S, where S is your entire sample space, okay? So if you're thinking of, you know, your standard, if you remember from school, your standard Venn diagrams, you have a box, the whole thing is your sample space, and you have events inside that you usually represent with circles. Remember that? Yeah. So these two properties together, saying that each event B1, B2, B3, these are non-zero probabilities and they are disjoint. That is called a partition of S. And then finally, your entire sample space S is a union of all these events, 1 to n, bk. Okay, sorry. So this is, these two make it a partition, okay, where you have these disjoint events, all of them together. If you take the union, you'll get the full sample space, okay? This is important. So if these three properties are satisfied, then you can show that for any event A, you can show that P of A is simply given by this quantity here. K is equal to 1 to n, P of Bk, P of A given Bk. Okay, so this is also very important, and it's called the law of total probability. So intuitively, what is going on here is very easy to understand. You have an entire sample space that you've broken up into chunks, right? These are the individual B1, B2, B3, B4, and so on, up to Bn. Now you're saying you want to ask what is the probability of some event A within that sample space. So you first say you condition on Bk, so your probability of A given that you are in Bk, and then probability of Bk, yeah? So you're sitting on the event Bk, right? So with some probability, and then you ask what is the probability of observing A given that you're already in Bk, okay? And then of course you have to sum over all the k's. Okay? So this should be quite intuitive. If there is any confusion, let me know. Yeah. So yes, so if you, so what you're asking is what the normalization is. So if you sum k is equal to 1 to n, p of bk, this is 1. That's the normalization. Yeah, that's over here. Is that okay? Any confusion here? Because really these are the only three things that we'll require to develop the theory of Markov chains. So if there's any confusion, let me know right now. Okay. So if that's okay, so we'll go on to now what are Markov chains. Okay. So let's start with a very classical example that all of you are familiar with, which is not a Markov chain. I mean, it's trivially a Markov chain, but it's not what you would use Markov chains for, is the example of a coin toss. Example of a coin tossing experiment. Okay. So let's say you have these random variables, x1. So what is a random variable? Some of you may not have seen this nomenclature. When I say x1 is a random variable, all I mean is when I repeat a certain experiment, for example, a coin tossing experiment, right? So let's say the toss of a coin is a random variable. What this means is every single time I toss a coin, the outcome is going to be different, right? So when it's a coin, the outcomes are head and tails, and each outcome has a certain probability, yeah? So that's essentially what a random variable is. Think of it as an experiment that you do. Every time you repeat this experiment, you get different outcomes. Okay, so that's a random variable. So now imagine here a set of n random variables. Okay, so let's say x1, x2, x3, all the way up to n. So I have n random variables, and I'm thinking of these random variables as coin tosses. Okay, so these are independent n random variables representing coin tosses. Okay, so one realization of this process, for example, could be heads, heads, tail, whatever, right? So on, tail. That could be one realization. So if I repeat this experiment, this is going to look different. Yeah? So now notice that these over here are independent. And independent in what sense? In this sense, that if I were to write down in this sense here, so if I were to write down the joint probability density of, or the joint probability of observing these n random variables in some state, they would simply be the product of each of the individual probabilities, right? Just the way it's written here. Okay? So this should be familiar with everyone. But in biology, examples such as this are very rare. Okay? So let me give you an example. So let's say we take the example of protein folding, for example. So once again, if I write down, say, n random variables, x1, x2, up to xn, and now I'm thinking of this index n as time. Okay? This is in fact the most common situation where Markov chains are used, where the index that you use to describe your random variables is time. Okay? So x1, x2, x3, x4 is a time series. Okay? So think of it like that. And I imagine these random variables to describe the folded state of a protein. If you have, for example, a small globular protein, then so you could have, let's say, you're thinking about protein folding, and generally small proteins tend to be in a, say, folded state or a slightly sloppier state, that's, let's say, the misfolded state, or a completely unfolded state. Okay, so imagine that your protein has these three states and it can transition between these states here. Okay, so now if I were to do some kind of time series to observe the folded state or the unfolded state of the protein, which is described by these random variables here, what you would almost always see, and of course this depends a little bit on the resolution of your experiments, but what you would see is something like this. You would see unfolded, 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 and it will stay unfolded for a while. Then maybe it will switch to the misfolded state. You'll see misfolded state for a while, and then maybe it ultimately lands up in the unfolded state. Okay? In this case, these random variables are not independent. Okay? See, these are not independent. Because if you've seen the protein to be in an unfolded state at this time, it's very likely that in the next time step, you'll also find it to be unfolded. Okay? This is very different from this scenario here, where this outcome has no bearing on this outcome here. But here, this is not the situation. Any questions? So how do we handle such non-independent data sets, right? That's where Markov chains come in. And of course, you make certain assumptions, and the simplest assumption one can make to handle this kind of correlated data set. And remember, why am I bringing this up? Because you can immediately make connections to this question of cell state transitions, right? You can imagine your cancer cells from, say, Yogesh's talk yesterday to be in a certain state, right? If it is one of these memory genes or memory states, you have the state for a while through many cell cycles, and then you switch to a different state, right? So therefore, you have correlations between different generations. And to handle that, we are going to use Markov chains. So Markov chains essentially is the simplest approach to modeling non-independent random variables, okay? Yeah. Yeah, so of course, I mean, it's a slightly more philosophical question because if you were to sample only very rarely, so once over here, then once somewhere here, of course, then it goes back to a problem like this, right? Because then everything is going to look independent. But on the kind of timescales that you make measurements in real biology, that is never the case, right? You generally tend to make measurements that are separated over short time scales. So these measurements then become not independent. Okay. So maybe hold on to that thought. You'll see how we are going to approach this, right? So then the question becomes, how do you deal with these kinds of correlated random variables? Clearly, the correlation lasts for a while, okay, over many of these realizations. So Markov chains are a very simple approximation where you say that given that you've observed the system in a certain state at one time point, your rest of your history doesn't matter, okay? So if I know, so this is where conditional probabilities are going to come in, and I'm going to formally write this down. If given that your system is in state U here, your next state is determined only by that and not by any of the history of your system. Okay, so that's a simplification, right? Because in principle, you should be writing down the joint probability of all these random variables together. But that's an impossible task. Hence, the simplest next step is to say, conditioned on a particular time and the state at a particular time, the rest of the history doesn't matter, okay? So formally, therefore, let's write down what a discrete time, and throughout this lecture, we are going to be thinking only of discrete time Markov chains, right? Where these indices are discrete, going from 1, 2, 3, 4, and so on, all the way up to n, right? So these are discrete times. So discrete time Markov chains. So let's write down a little bit more formally what a discrete time Markov chain therefore is. So you say you consider the random process. Once again, random process is a technical term. It just means a collection of random variables. Okay, that's sufficient for this class here. A random process just means a collection of random variables, x1 up to xn. Okay? So consider the random process, the kind of things that we've been looking at, xn, where n is 0, 1, 2, up to n. Okay? Where the range of xi, which is also called the state space, S belonging to 0, 1, 2 up to say R. So notice there are two important distinctions over here, okay? One is the state space and one is the index that, that denotes the random variable. So here, x1, x2, x3 up to xn, these are n random variables, yeah? Each random variable, for example, x1, can go into three different states, right? Folded, misfolded, unfolded. So the state space here is three different states, F, M, and U. So that's the state space. So the state space and the index are two different things. So don't confuse these two things, okay? So we're talking about a random process, Xn, which is the index of the random variable, which will mostly denote time, right? So n is 0, 1, 2, 3, all the way up to n. And the range of each of these xi's, right? Each of these random variables, which is the state space, is denoted by S, okay? So for this example, the state space is 3. You have 3 states. Okay, so in this process, is a Markov chain if the following holds: P x m plus 1 is in the state J given xm equal to I, x m minus 1 is I m minus 1, or so on, all the way up to x0 equals to I0, is equal to P x m plus 1 equal to J given xm equal to I. For all m, j, I, I0, all the way up to I m minus 1. So let me pause and see if you understand this. So remember, the intuition I gave was that conditioned on a particular time, none of the history matters, right? So that's all that I've written over here, that the x given xm equal to I, so this is the conditional probability of your m plus 1th time step to have the state J given your entire history, okay? This is m, m minus 1, m minus 2, all the way up to 0, okay? So this is your entire history. I'm saying if this entire history doesn't matter, but only the earlier time step matters, that's what I've written here, right? So if this entire quantity with all of this history, if this is equal to this probability here, where the m plus 1th time step is in state J, depending only on the previous time step, that is called a Markov chain. Is that okay? This has the entire history. I'm simplifying that and saying none of that history matters. It's only the earlier time step that matters. So if I know that my protein was unfolded here, only that matters for what the state of the protein will be in the next time step. Okay? You could have imagined, and there are non-Markovian models where all of this history matters. Okay? But that, of course, becomes much more complicated. Okay. So a few things about nomenclature. So these are important things to remember so that you understand Sahan's paper better. because the Sahan's paper has a particular form of the Markov chain. So certain things to remember is if the number of states in my state space, states in S is finite, then this process is called a finite Markov chain. Okay, finite Markov chain. So all the examples that we've discussed here, where your protein has 3 states, the coin has 2 states, these are all examples of finite Markov chains. Okay? You can have examples where this is a continuous variable and hence there are infinite states. Okay, so we are not dealing with, so what we are going to deal with to understand that cell systems paper is a discrete time finite Markov chain. Okay, so that's one. The second thing which is very important is this quantity here, right? This quantity is called the transition probability. Okay, so P xm plus 1 equal to j given xm equal to I, this is called the transition probability. So these are transition probabilities. And we are going to make an important assumption throughout, and this is in fact the most common assumption to make, is that these transition probabilities do not depend on the index m. So these are time independent. Okay? So P's are not dependent on m, hence we write these things as just P of, say, ij. Okay? So the transition probability of going from state I to state j, irrespective of which time you are looking at. Okay? So if I'm here, what is the probability that I'll transition to a folded state in the next step is independent of whether you're looking here, 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 and so on. Similarly, if I'm here, the probability of transitioning into a different state is independent of whether I measure it here, 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 or here. Okay? So that's the assumption that is usually made in these kinds of Markov chain processes. Okay? And finally, the important thing for building up the mathematics is that usually these transition probabilities are clubbed together into what is called a stochastic matrix. Okay, and the properties of a stochastic matrix are very fascinating, and it comes back to discussions we were having earlier with Herbie about the properties of eigenvalues, eigenvectors, etc. of these matrices. So this is a very important idea, that of the transition probability matrix, which is just clubbing all these probabilities together. And so throughout, I'm going to denote the transition probability matrix with this P with a tilde. Okay, so this implies that this is a matrix. So P, so let's say you have R states in your state space. So you have state 1, state 2, all the way up to state R, and you're making transitions into states 1, 2, all the way up to R. Okay, so that's how you read this matrix. So you are in state 1, you are transitioning to 1 or 2 or 3 or R. Okay, so these are P11, P12, all the way up to P1R, and similarly PR1 all the way up to PRR. Depends on how many states, right? So if you're looking at this probability of protein folding, this will just be a 3x3 matrix. Yeah, going from unfolded to unfolded, unfolded to misfolded, unfolded to folded, and so on. Okay. So can someone tell me what is the normalization condition for matrices like this? How would I write the normalization? Yeah. Perfect. So sum, of course, right? Probabilities have to add up to 1. So sum of each row has to add up to 1. Okay. So for all I, sum over k equal to 1 to r over all the states, p I k is equal to 1. Okay. So all rows add to 1. Okay. So this kind of a matrix where all the rows add up to 1 are called stochastic matrices. And they have some really fascinating properties which we'll get to by the end of this class. So I have a feeling we may not be able to get to Sahan's paper. So we'll see, maybe some other time that's free I can finish that paper. But today it's important to go through the theory very carefully, right? So I'll still go a bit slow. Okay, so one important point to note for all students is that in the literature, you will often see a variant of this, okay, where the rows add to 0. Okay, these are called transition rate matrices. So you should not confuse the two. These two are related. When you see a matrix like this, but the rows are adding up to 0, rows are adding up to, sorry, here it's 1. So here the rows add to 0. These are transition rates, okay? So don't confuse the two. This is transition probabilities. These are real probabilities, right? So these numbers have to be between 0 and 1. But there is a version of writing down the same kind of Markov chains with transition rates, which are probabilities per unit time. That's a slightly different formulation. It's exactly equivalent, but there the rows will add up to 0. Okay, so many books will do it that way. So don't get confused. No, they can't be negative rates, but what happens is by definition, the diagonals are set to negatives. So I didn't want to bring that up because it's a bit confusing. So the diagonals will be the negative of the sum of the rest of the rows. Okay, so that's how transition rate matrices are defined. Okay, so that's why I didn't want to bring that up, but if you are reading any books, there are these two alternate versions that you'll see. So don't confuse it. We'll stick to, so then what happens is the eigenvalues also change. There are theorems that I'm going to talk about where it gets confusing. So remember that we are sticking to this nomenclature. Okay. Okay, so I'm now going to show you a simple example because this is actually quite a lot. So let's see if you can work through a couple of simple examples that will help sort of establish these ideas in your head and how to actually work with Markov chains. Okay, so if you have a good sense of that, Sahan's paper should be then easy to figure out. Okay, all right. So stochastic matrices, we'll come to the properties of stochastic matrices in just a bit, but I want you to solve a simple example. And I'll solve one, and then I'll give you something to solve. Okay. So let's take a simple example to see how these Markov chains work. Example. So let's say once again we have a 3-state Markov chain, just the way we've been thinking about protein folding. Okay, so we'll keep it simple. It's a 3-state Markov chain. So we have 3 states, A, B, and C. Okay, so my state space is 3. Think about this as, say, unfolded, misfolded, folded. Yeah. This already is actually a very nice example because you have real research that uses this kind of formulation to think about the protein folding problem. In fact, if you're interested, I have a paper. I didn't want to mention it, but it's okay. You can take a look. It's exactly on these kinds of things. Et al. It's in PNS. I forget, maybe 2015 or something. This has to do with chaperone unfolding of proteins. You can take a look after this class. It's exactly this problem here with exactly the same formulation. Okay, so you should be able to follow it completely. So you see that various different kinds of problems can be solved with similar frameworks. Okay, so now I have to define my transition probabilities. So let's just put some numbers here. Let's say this is half. This is, let's say, 1/3. This is, say, 1/4. Let's say this transition is, say, 1/4. This is, say, 1/2. This, I'll say, is 2/3, and this is half. Okay. So these are the two main things for defining a Markov chain, right? You have to define your state space, and this, as we've been seeing, depends on the biological problem of interest usually. So here we have 3 states, and then the important thing is to define all the transition probabilities between all these states, right? So then your transition probability matrix, my stochastic matrix, will look like this, right? So you have the 3 states, ABC. So let's fill that out. A to A, what's that probability? 1/4, right? A to B, half. A to C is 1/4. B to A is 1/3. What is B to B? 0, good. I'll just fill this out. Half, 0, half. Okay? All right. So what kind of computations are we going to do with systems like this? So let's take a very simple example and let's try to solve this, right? So the question is, okay, so let's work through this problem, and the next problem you will solve, okay? So if I say that if P of x0, remember the indices always refer to time, right? So if P of x0 equal to A is 1/3, I want you to find P of x0 is equal to A and x1 equal to B. So what's the problem? I'm saying you have a system like this. You have 3 discrete states with all these transition probabilities. This is my transition probability matrix. I have set up all the theory to start answering questions like this, which is at time 0, I have told you that the probability of my system to be in state A is 1/3, okay? I have to give you some initial conditions, right? In any kind of dynamical system, you need some initial conditions. So the initial condition here is that at time 0, I am in state A with probability 1/3. Now I want you to tell me what's the joint probability of being in state A at time 0 and in state B at time 1, right? In the next time step. That's the question, okay? We are slowly building up towards understanding. Remember, the end goal is to understand that cell systems paper. So these are the kinds of computations that are required to understand that ultimate paper, yeah? So anyone, can you tell me how would I go about doing this? Remember we set up 3 things in the beginning: conditional probability, the chain rule, and the law of total probability. So just using that, you should be able to solve this. So let me do this so that you can try the next problem. So how would I do this? Remember, I have to break this. So very obviously, I have to break this down and write it in terms of conditional probabilities, because that's the kind of stuff we have in the transition probabilities. So what I would do is, I'd say this joint distribution is, can someone tell me how should I write it? X naught equal to A, P of what? Okay, very good. Very good. Yeah. And so what are these 2 quantities? What are the numbers? This is 1/3, right? Because that's what I've given you. That's the initial condition. And what is this? This is just P of 1 going to 2, right? This conditional probability is just, this is just P of AB, right? And that is P of AB is half, so this is just 1/6, okay? So starting from this initial condition, we know how to write down probabilities for the next step, okay? So why don't you try this problem now? It's slightly harder. So I'll give you the same initial condition. If P of x0 is equal to A is 1/3, so same initial condition. Now find P of x0 equal to A, x1 equal to B, x2 equal to C. So now I have 3 variables. At time 0 is A, time 1 you switch to B, at time 2 you've switched to C. Try finding this. So maybe take 3 or 4 minutes and give me a number. Hey, Mohit. So I'm definitely not finishing today. So maybe I'll finish Markov chains. Maybe some other day I can just take 15-20 minutes tomorrow afternoon, whenever, and then I'll show them how to derive Sahan's paper. So that after, yeah, just 15 minutes should be enough. Who can you come maybe and work this out? Yes, it is 1/9. Very nice. So yeah, please. Where is the? No. Maybe we'll wait for 2 minutes and give the others a chance to solve it. So maybe we wait for 2 minutes and then we can solve it. Has anyone else managed to get 1/9? Okay, some of you are getting it. Will you solve this? Thank you. So if you could also walk everyone through what you're doing, that will be great. So that's just the chain rule, right? If you remember, this is equation 2 that we had written down. That's the chain rule. So that's just using the Markov property, right? So the last term, the conditional part becomes conditioned on only the previous step. And then we have all these numbers here. 1/3, I think. Thank you very much. Yeah. I think next problem should be harder, I think. Okay, great. So now you have a sense of how these Markov chains work, right? So so we'll formalize this a little bit more and see how, oh, even this is quite a bit. So you've seen the basic math of how you evolve these systems. So we'll formalize this a little bit more and write down what are called the state probability distributions now. Okay. So essentially what we are given is, suppose we are given the following. So just like before, you need an initial condition, right? So I'm going to now write this down in complete generality, right? Remember here, I was just giving you the initial condition for one particular state, that at time 0, the probability of being in state A is 1/3. But at time 0, the other states also have some probabilities, right? So now I'm going to generalize this completely and write down the full general evolution equation for Markov chains. So the state vector of probabilities I will denote as pi, and the dash on top will denote a vector, yeah? And this, what is it called? Superscript is now going to denote my times, okay? So it's a bit confusing, but now I'm going to write this as superscripts, okay? So pi 0, which is my initial condition, so this is then just my initial condition. Initial condition is just my vector of probabilities, right? So this is x0 is a probability of x. So now I'm going to switch from that example and I'm going to use 1, 2, 3, 4, okay? Sorry, this might be a bit confusing. So let me define this clearly so there's no confusion. So my Markov chain is xn, n going from 0, 1, and so on. Right? So these are my indices, the times, and then my state space here now, I've gone back to S is 1, 2, all the way up to R. Okay? In the previous case, I was using state space as A, B, C, D for that example, but now I'm going back to 1, 2, 3, 4 to generalize this problem. Okay? So x0 equal to 2, and so on, up to you have x0 equal to R. Okay. So this now is my initial condition. This is the vector of all the initial probabilities. Okay, so now this has become a vector because you have R states, and I want to give you the probabilities of all the R states. So the normalization is just that these add up to 1. Yeah? Okay, so what we want is to ask, how do you find pi n? Okay, so you're given pi of 0, that is the vector of probabilities of your initial time. At any arbitrary time n, what is my vector of probabilities of finding the system in any of these R states? So that's the question, okay? So it'll just take 1 minute to get this. Sorry, is there a question? Wait, who's asking this question? Yes, it will be pi naught into p to the power. Yeah, but we'll see how that comes. So that's my question. How do I solve that? Basically, we will use the law of total probability. Law of total probability. So for any state J belonging to S, I can write down P of x1. Okay, so let's write down the first time step, and then we'll see that this, you'll see an obvious pattern. P of x1 is equal to J, right, is simply this sum here. K is 1 to R, P of x1 equal to J, given x naught equal to K, P of x naught equal to K. So I've used equation 3. Using equation 3, which was my law of total probability. So what have I done? Once again, this should be quite intuitive. At time 0, I have some probability of being in state K. From state K, I want to jump to state J. That's the probability I'm trying to solve. At time step 1, what is the probability of being in state J? This could have occurred from any of the states at time 0, right? So then I have to sum over all the states. Yeah? So this is nothing but sum over K is equal to 1 to R, P, Kj, P of x0 equal to K. Right? So this is P of x0 equal to K. That's where you're starting from. You're jumping to the state J from K, and K can be any of these states, so you have to sum over this. Okay? Now if you generalize this, you'll see, and I leave this for you to convince yourself, it's a one-line thing, generalizing, you'll see that pi of 1, that's at the first step, is just pi of 0 multiplied by the transition probability matrix. So this term here, if you just check this, you can write this down as the probability, the matrix multiplication, right? So this is a vector, this is a row vector, and this is a, so this is a row vector which has dimensions 1 cross R, and this is R cross R. Okay, so ultimately you end up with a 1 cross R vector, which is the vector of probabilities at time step 1. Okay, just check this by writing out the matrices. It's really trivial. Okay, so now that you have this, Mohit, can I take 5 extra minutes? Yeah, we have coffee break after this, right? We are almost done. The fascinating limiting distributions I'll come to in just a bit. It won't take more than 5 minutes. Okay, so check this, write out the P matrix as P11, P22, the way we had written it down. Multiply this out, take the sum, you'll see that this is exactly going to be this, right? So instead of breaking it down with indices, I'm just doing it as a matrix multiplication. Okay, so now if pi 1 is pi 0 into P, it's easy to write down what pi 2 will be. So can someone tell me what will pi 2 be? Right, good. So it will be pi 1 into P. But pi 1 we've just written is this. So I can put this down and it will become pi 0 into P square, right? And so you see the structure here. What you'll ultimately get is at the nth step, you have pi 0 p to the power n. Okay, so that's your most general way of advancing your Markov chain. You start with an initial state probability vector and you just multiply it with the transition matrix, the stochastic matrix, to the power n, okay, where n is how many time steps later you want to see where your state is, okay? All right, so something really nice comes out of the properties of stochastic matrices, and we'll cover that in a couple of minutes, which is the limiting distributions of these kinds of Markov chains. This also becomes important for understanding Sahan's paper. Okay, so limiting distribution. What do I mean by limiting distribution? It just means what would you expect when n tends to infinity, okay? So the question is, what does pi of n look like when n tends to infinity? Okay, so if you've waited long enough, which is usually not too hard to achieve in biological experiments of this nature, what would you expect pi n to look like? Okay, that's the question we are asking. Something I wanted to say here. Okay, let's see. Right. What I wanted to say is it's very easy for you to check this on the computer and convince yourself that there really exists limiting distributions, right? So you can take any simple example. For example, in this example that we were looking at, right, set up some initial condition. For example, set up something like this. I don't know. You have 3 states here. So you can just start with 1, 0, 0, meaning all your probability exists to be in state A, right? Set this to, for example, 0 and 0, all probability in state A. Then try, just on your computer, do this matrix multiplication. Pi 1 is just this pi 0 into p. Yeah? Keep doing this for many times. You'll see that within 5-10 iterations, you can do this over your coffee break. You will see that this quantity will reach some numbers. So for example, if you do this, what will be the next step, right? You will just have to do this matrix multiplication, right? 1, 0, 0 multiplied by this matrix here, right? If you do that, let's say you get some numbers. I'm just making this up. Say half, 1/3, 2/3, right? You keep doing this, you'll see that initially these numbers will change, but after a bit, these numbers will become fixed, okay? They will not change with time, meaning you've reached a stationary distribution, okay? So how do you prove? So you can in fact prove that systems like this must reach a stationary state, okay? So let's see how you do that. So to do this, we will need to resort to the ideas of eigenvalues and eigenvectors. So I hope all of you have seen eigenvalues and eigenvectors before. So what we'll just say is let lambda I and pi I. So don't get confused by the notation. The, what is this, subscript, right? So the subscript here refers to eigenvectors. That's why I was keeping these as superscripts to indicate time over here. These are going to be eigenvectors. So let lambda I and pi I be the ith eigenvalue and eigenvector of the stochastic matrix P. Okay? Now remember, P is an R cross R matrix. So to keep life simple, we are going to say that you have R independent eigenvectors that you can get out of it. It gets more complicated when these are not independent, but for the moment we are going to just think of it like that. So because lambda i's and pi i's are the eigenvalues and eigenvectors, I can therefore write down the eigenvalue equation pi I for any general I. P is just lambda I pi I, right? That's my eigenvalue equation. That's my standard eigenvalue equation. And so if I right multiply with this P over here, pi I get P. I'm just doing matrix multiplications. I'm going to get lambda I pi I P. Now what is the right hand side? Can someone tell me? Right, very good, because this again, you can write this as the eigenvalue equation. So this just becomes lambda I, pi I P will become another lambda I times P. Oh, sorry, pi I. Okay? Is this clear to everyone? I see some confused faces. I'm just using the fact that I have an R cross R matrix here, P. It will have R eigenvalues and R eigenvectors. So any one of those eigenvectors I'm indexing by I, right? And then I'm writing down the eigenvector eigenvalue equation. Pi I P is lambda I pi I. This is the standard eigenvalue equation for the matrix P. And then I'm just using the same property over and over again. And basically what I'll get is pi I Pn is lambda I n pi I. Okay? This is equation 5. Okay, I'm just repeating this over and over again. I'm right multiplying with P. So n times, if I do it, pi I P to the power n is just lambda n pi I. Is that okay? So now I'm going to assume that I have a basis set. So this is an assumption. It may not always hold. So assume the R eigenvectors form a basis, meaning that any vector in this R dimensional space I can write down as a linear combination of the eigenvectors of that P matrix. Okay, so that's what I mean by a basis, and therefore what I'm going to do is even the computer wants a coffee break, I think. So what I'm going to do is I'm going to write down the initial vector. Remember, this is my initial times 0 vector. I'm going to expand this using the basis vectors of P. Okay, so C1 pi 1 plus C2 pi 2 and so on, Cn pi n. Okay? Remember, I've assumed that the R eigenvectors sorry, if there are R eigenvectors, I should write R here, right? So just go back and make sure that my notation makes sense. Okay? I may have mixed up some of my R's and N's. I was doing this last night. Okay, but anyway, you'll get the point. So from 4, now, what I can do is pi n this quantity here, right? I'm going to use this is pi 0 p n, right? That's just equation 4, and oops. So this now, I'm going to substitute the expansion. So I have C1 pi 1 all the way up to CR pi r, and then I have Pn. And now notice that this is exactly in the form of this equation 5 that we had written, right? You have pi 1 multiplied by p to the power n, right? So now this is very nice because pi n I can now write down in the most general form as C1 lambda 1 to the power n pi 1 plus C2 lambda 2 to the power n pi 2. I'm not supposed to cross this apparently, but it's okay. Right. Right, and all the way up to CR lambda r to the power n pi n. Okay? Pi r. And this is, there are some dots, CR lambda r to the power n pi r. Very good, some of you are awake. Okay? So this is now my most general expansion of the 2 minutes of the state vector at time n. Okay, so notice I've written this down in the form of eigenvalues and eigenvectors of the P matrix, and this is where two properties become very important. So one is the fact that for any stochastic matrix, okay, for any stochastic matrix P, you can show that these lambda i's have to be less than equal to 1, okay, these lambdas. These lambdas are my eigenvalues of the P matrix, and you can show, and this comes from a very beautiful theorem which is called the Gersh-Gorin circle theorem. So usually I do this in class, but I think you'll beat me up.
If many of the lambdas are greater than 1, this thing is going to blow up, and it can satisfy the conservation probability.
No, but I mean, how this is for any matrix with these sums up to 1, the rows, if they have to sum up to 1, I don't think it's that evident that the eigenvalues have to be less than 1.
Well, if you look at your expression, pi n is going to have elements which are growing exponentially if lambda is greater than 1.
Right.
And therefore, pi n has to be normalized. The sums of the pi n's have to be 1. And therefore, no term in that expansion can be growing exponentially.
Let me think about it. No, good point. I mean, you make a good point. I haven't thought about it. Let me think about it. No, but I mean, what Herbie is saying is, why do you even need to have a theorem to say that the eigenvalues have to be less than equal to 1? Because otherwise this would blow up. Let me think about it. Yeah, I haven't. Yeah, good point. I haven't thought about it.
I teach this also.
Right. Right. No, but there is something, because is it guaranteed that p to the power n, there the rows will also be normalized. That's true. Yeah, let me think about this. Yeah, I think it's worth thinking about this, because usually you generally try to show that this is less than equal to 1. Okay, but what about so the second one might be more convincing, which is that at least one eigenvalue has to be exactly equal to 1. Right? Exactly. The row sum is 1, and this comes from the fact that you can just take the P matrix, multiply it with a column that is all ones, and that will be 1 into 1, right? Because the row sums are all equal to 1. So there has to be at least one eigenvalue that is equal to 1.
You missed the part of it here. If you're being rigorous, you need to say that left eigenvalues.
Yeah, left eigenvalues, right eigenvalues. Yeah. I'm already, people are going to kill me for not letting them go for coffee. Yeah. So there is all the left eigenvalues, right eigenvalues, which I'm not going into. But yeah, this basically is the final step. So you can see that, where shall we go? Here. Yeah. Sorry, say that again. Yeah. No, because the point is that as long as you have R distinct eigenvectors, you can write down any vector in that R dimensional space as a linear combination of these eigenvectors. So it doesn't matter what initial vector you start with, right? As long as it's R dimensional, you should be able to write it. Okay, so then once you have that, of course, as n tends to infinity, what will happen is, notice that since all your eigenvalues are less than equal to 1, each of these eigenvalues to the power n will all become 0, right? So each of these terms will go to 0, except the one eigenvalue that is exactly equal to 1, okay? And therefore, you just have the following. As n tends to infinity, your stationary state, if I denote it by that, which is just the limit n tends to infinity, pi n, will just be C1 pi 1, okay? So this pi 1 is the eigenvector corresponding to the eigenvalue 1, okay? So what you've shown is that no matter what your transition probability matrix looks like, as long as it's a stochastic matrix where the rows sum up to 1, you have to have at least one non-trivial steady state as n tends to infinity, okay? So you will always have a steady state. It's not that you may or may not. You will always have a steady state. Okay, I think we'll stop here, and maybe someday, maybe tomorrow or day after, we have lots of time, I'll finally show you the connection between all of this and Sahan's paper, yeah? Okay, so I think we'll take a break.